So today's award is the Alfred Redfield Lifetime Achievement Award, which honors the career achievements of an aquatic scientist whose work is recognized for its importance and long-term influence. This year we're honoring Bruce J. Peterson for, well, I'll quote, innovative and transformation studies of carbon, nutrient, and water cycles at process, ecosystem, and global scales. Trying to figure out what to say about Bruce is difficult. You can't point to any level of science that he hasn't had an influence in. He's been influential in developing our understanding of mass flux in the oceans in terms of particle flux, developing new techniques such as multiple stable isotopes. One of his papers published in Science in 1985 was one that actually now has ended up driving my research for almost 20 years. He's been involved with the application of whole ecosystem chemical labels, and he's developed a much more comprehensive understanding of the hydrologic control, or the controls of hydrologic budget of the northern hemisphere, particularly in the oceanic domain. So Bruce basically can do it all, and as part of his nomination letter, it was pointed out that he's a remarkable synthesizer, and I thought that maybe that English wasn't right on that, because I thought, was he in a band at one point? He wasn't. He's a synthesist. He's got incredible insights on a bunch of different topics, and in talking with him briefly last night, he said something that resonated immensely with me, which was that the key to this sort of big scale synthetic collaboration is trusting your partners, letting your guard down, letting people have an influence on your research. And I think all the way through his career, Bruce has shown that he's capable of doing this, taking the best out of everybody, bringing it together with the best of his insights, and creating science that is transformative at all sorts of different scales. So with that, I'd like you to help me welcome Bruce, the 2013 recipient of the A.C. Redfield Lifetime Award, Lifetime Achievement Award.
Well, this is all a little overwhelming. I'm going to tell a story about how I got to Woods Hole, how I got involved in these studies, thumbnail sketches of a few of the projects I've been involved in, and end on a note about collaborations. Let me see if I can advance this. Probably none of you know that Redfield is my scientific grandfather, that he was the advisor to my major professor, John Barlow, who was at Cornell. I have employed Redfield ratios, that is the ratios of C to N and P in the oceans of the world, throughout my career. And for the past decade, I've lectured on the Redfield hypothesis in our semester in environmental science course at the Ecosystem Center in Woods Hole. However, receiving the Redfield Award is a complete surprise. I had no idea I had been nominated, did not know anything about it until John Dowling called me. But because of my relationship to Redfield, for me, it's a very, very special honor. At the beginning, Alfred Redfield on the left with a collie. I'm on the right, bathed in a cloud of dirt. And my mother was horrified to find out that my pets were in my pocket. I had worms in my pockets. we didn't have a TV in my home. I spent my time outdoors. Here I am with a snake, feeding a fawn. I believe very strongly that the time I spent outdoors influenced my career very profoundly. There were a lot of other influences along the way. In high school, I had a biology teacher who looked like a big grasshopper with bug eyes, but inspired me in ways that you can hardly imagine about biology. I had a predisposition, I think, to be excited by biology. I went to Bates College and I was a biology major. And of course, if you're a biology major at Bates College, you're expected to be an MD. In my junior year, John Canwisher from the Woods Hole Oceanographic Institution visited Bates and gave a lecture on the ecology of salt marsh trace gas fluxes. Well, that gave me a new model for my career, influenced my decision to go on to Cornell University and to major in ecology and systematics. You know, I always believe that if there's a fork in the road, you should take it. I actually applied to the fisheries science department. They had run out of fellowships, and some very kind professor, John Barlow, picked up my resume and invited me to join the Department of Ecology and Systematics. So another turning point. I got my PhD and I decided that I wanted to go into research full-time. I heard that George Woodwell was going from Brookhaven to Woods Hole to found an organization called the Ecosystem Center. So I wrote to him begging for some sort of a job. I got no reply. He doesn't even know to this day that I actually did that. However, I went to North Carolina State University, had a postdoc with John Hobby in 1975. It took all of about 6 months before Hobby walked into my office and said, "I'm leaving." "Where are you going?" "Well, I'm going to the Ecosystem Center in Woods Hole." I said, "So am I." So I got in, and I was promised that I should not expect more than 1 or 2 years. They didn't know how to get rid of me because they found out I could write grants. However, I had some big decisions to make now about how I was going to proceed to understand how ecosystems work. I had spent a PhD thesis measuring processes in bottles. I'd become increasingly skeptical about understanding ecosystems with these piecemeal and artificial approaches, especially after my thesis paper was rejected by LNO. So what were the alternatives? Well, I could do whole ecosystem perturbation experiments, messing with ecosystems, or I could be more clever, do whole ecosystem isotope tracer studies, and I've done both, and I'm going to give you some examples. However, first, here's a list of the major mentors and collaborators that I've worked with over the years. My mentors I've mentioned already: John Barlow at Cornell, a student of Alfred Redfield, John Hobby, and George Woodwell. I've had a large number of collaborators. I'm not going to read all their names. They fall into 3 categories. I've studied streams and rivers, I've studied estuaries, and I've worked on various models of the global biogeochemical cycles over the years, and that was, of course, inspired by George Woodwell. I'm going to say a couple slides, just a little thumbnail sketch of some of these studies. This is a talk not so much about the science, but to give you the gist of what this work was all about. Well, the first paper I'm going to talk about is Eppley and Peterson, 1979, Particle Flux and New Production in the Deep Ocean. This was a collaboration that was a complete serendipitous thing. Dick Eppley got a proposal on the global carbon budget. I had written an addendum to the proposal using various biogeochemical models to calculate the sinking export flux of particles from the surface to deep ocean. I was trying to look for where's the missing carbon in our understanding of the global carbon cycle. Well, Dick got the proposal, threw the proposal away, ripped off my addendum, and realized that it was complementary to the work he'd been doing that was pioneered by Dick Dugdale, using 15N tracers of nitrate uptake and comparing that to the regenerated production, that is the reduced forms of nitrogen recycling in the surface layers of the ocean, comparing that to total production and calculating from that what the export production should be. So by combining these two different approaches, and you understand the Redfield ratios are right at the heart of this again, we were able to get this paper published. And this was the Martin Award paper several years ago, and Dick, I know, talked about this. I won't go into it anymore, except to say that I was disturbed that turns out the Martin Award isn't to the authors. It belongs to the paper itself. So it's the paper that gets the award. So I'm back again. This is the Kuparuk River on the north slope of Alaska. Anyone who loves the outdoors would be thrilled to spend, 37 years working their summers on the north slope of Alaska. This is the Kuparuk River. We did a 30-year, this would be the 30th year of polluting the stream with phosphoric acid. We just drip it in. And it's a low-level perturbation, 0.3 micromole of phosphate. It's not a lot of phosphorus. But the natural level of phosphorus in this system is almost undetectable. So this is about 10-fold greater than the natural level. The major thing that happened, a lot of small changes occurred very quickly, but the major thing that happened is illustrated here. We started out on the left with a rocky bottom stream, bare rock. It took about 7 or 8 years for this cover of moss. It's a nutriophilic moss, Hygrohypnum, that was not present in the reach when we started the experiment. Now this is sort of analogous to a lake gaining macrophyte beds, or an estuary having or not having seagrasses. It changed everything about this ecosystem. Its food webs, every insect species was affected. The biogeochemistry, of course, is greatly affected. I looked at that and I said, well, I want a way of, in a quantitative sense, of comparing these two stream reaches and with respect to biogeochemistry and food webs. And so we developed the idea that if you could add 15N ammonium as a tracer, it's very hard to point from here. I'm going to try here. Add that. It would only take a few grams of 15N to enrich this ammonium pool to about 1,000 per mil, which is a big enrichment, easy to measure on a mass spec. The ammonium then would be taken up by the benthos, nitrified, produced nitrate. The nitrate would subsequently be taken up. And this biota box here looks like this in the Kuparuk River. It is in fact an entire benthic food web. So if you have an idea that this is where your add the ammonium here, and you're thinking now about how would a filter feeder, such as a black fly, respond to this. And so the game is for you to figure out what the pathway is and how much of that nitrogen is going to go from here to the diatoms, and then slough off into PON, which is in transport, moving downstream and removed by them. Well, that transformation includes about 4 or 5 things that we have no way of measuring in streams, or we had no way of measuring. However, we had a little bookkeeping model that you could test your ability to model this cycle. And the data here are distance here, kilometers down the river, and the amount of 15N enrichment in the black flies. Data are the black points. The open points are the model. Well, of course, that's not a bad fit. That's, I believe, an 80% level of prediction. And so that's pretty good, but it probably took us 4 or 5 times. And to solve this whole matrix, let's see if I can go backwards. Can I go backwards? Yeah, to solve this matrix, oh, I went backwards twice, I'm sorry. Must be a lag in the system. Okay, to solve this entire matrix, it took several years of iteratively trying to figure out what these fluxes were. But for the first time, we actually could constrain many of these fluxes by following this isotope through the system. And if you're interested, the genius behind doing that was Will Wollheim, published in Jane Abbs, 1999. That study proved to be a virus. It was attractive to stream people from throughout the U.S. And at a LTER, All Scientists meeting, we got a working group together, a set of hydrologists in one group, biogeochemists in another, and decided we wanted to repeat this experiment throughout the country. And that is repeat exactly what we just described, the adding labeled ammonium. That was a great success. And so we decided to do a subsequent study where the trick was now to add nitrate to the stream. The idea here was to emphasize denitrification and the way that stream networks modulate the flow of nitrogen downstream and develop the models needed to predict how that would work. One advantage of labeling the nitrate is a 20,000 per mil, for those of you who know anything about isotopes, was to also label the N2O and the N2 so we could get the actual rate of denitrification and gaseous loss from the system. So that experiment that we had run on the Kuparuk River has now been run worldwide. And it has led to the 3 papers, one on retention of nitrogen, one by Mulholland on the denitrification, and one by Bilio on the trace gas flux, N2O, from these systems. And I was surprised to go to New Zealand about a decade ago and find out there was a graduate student doing exactly this experiment to look at the impact of imported fishes on streams in New Zealand. Okay, I want to switch and say something about the saltwater side of things. Natural abundance studies work well in estuaries because of the diversity of sources of organic matter. Land, marsh, and ocean have very different carbon, nitrogen, and sulfur isotope compositions. So natural abundance stable isotopes are very powerful. Added tracers are rarely, if ever, attempted because of the scale of these systems and the tidal dispersion losses, meaning that you can't afford enough isotope. So here's an example that Peter mentioned about the using multiple stable isotopes and looking at a salt marsh system where this is Buzzards Bay, and these numbers are locations in the marsh where we collected a filter feeder. And what we discovered is that the filter feeder had stable isotope ratios for sulfur that were a function of its location in the marsh, which sort of messed us up because we were always thinking that organisms and ecosystems would have a stable isotope ratio, somewhat predictable, and I could just go out and collect. Well, this disabused us of that notion. It's much more complicated than that. So what's the answer in terms of the sources? Well, if you would look at a plot of carbon here versus sulfur isotope abundance here, upland plants, Spartina, plankton, are very well separated on this plot. If you would just use the carbon isotope, you could combine that, and plankton fall in the middle, you can see you couldn't separate these three sources. But here, the Spartina end member is populated by the mussels that are back in the marsh, and as you move toward the Buzzards Bay and the planktonic system, these ratios change and exactly point to the planktonic end member. Well, I tried and failed 3 times at NSF to apply the multiple isotopic tracer to a riverine estuary where it would be much more powerful because they actually had all 3 end members. And Brian Fry and I worked on that, and we couldn't get it funded. So finally decided we would attempt just a 15N tracer study in an estuary. And this is the Parker River estuary in northern Massachusetts. The end member here, this is a freshwater end member, meandering stream. In the middle of this, we just put a dripper and just dripped in 15N nitrate tracer levels, so we're not changing the abundance of nitrate at all. And in the summer, this reach here flushes slowly enough that it develops a large bloom of a planktonic diatom. And so the issue was, well, what's happening to the nitrate coming from land as it traverses this reach that's very active during the season of the year? Here's what the stream looks like. That's Max Holmes, a postdoc in the boat. This is Chris Thulke, who did many of the isotopic measurements, working with Brian Fry. Big tidal range, about 3.5 meters or so. So this is mid-tide, and the tides would go up to the level here and flood the marsh at the very high tide. We were able to do a budget that showed that of the 3 kilograms of nitrogen per day, this is summer low flow, the actual phytoplankton uptake was about an order of magnitude greater than that. These phytoplankton diatoms were settling rapidly. They were dividing about almost once a day. There was a regeneration of inorganic nitrogen in the system, some recycling in the zooplankton in the system, and an export of about 3 kilograms per day. Well, that's a pretty dynamic system. It's churning over, and this is the flow through tidal estuary. It had a food web that is a little more complicated than the Kuparuk River. The planktonic portion is shown in the upper part here, where the top, that's the phytoplankton becoming labeled. You see how rapidly they become labeled from no label up to that. Then there's the planktonic copepods, the alewives, and the white perch, all part of the planktonic system. Surprisingly, the benthic components of the system, 3 of them were clearly linked to the sedimentation of the diatoms. They increased rapidly. They gained just about as much label as some of the components of the planktonic part of the system. But the isotope showed us there was a second food web. There was benthic diatoms. They only became labeled by about 15-20% as richly as these, but they were feeding the alligachetes, the benthicopods, and the mummy chugs. And then we had a portion of the food web that was not closely tied to this addition at all. Large mummy chubs were actually feeding up on the marsh at the very highest tides, and material that was not well labeled in the experiment. Let me say one last thing about this experiment. These sorts of experiments are, to me, very powerful. And if I had become that doctor, I think it would be perhaps the difference between the doctor having an X-ray and then finding that if he had an MRI and could look at it sequentially, you could learn a lot more about the system. So when I get information like this and we develop the models, I begin to feel that I've got my finger on the pulse of the system rather than rather sterile data, which are just numbers of biomass of each component over time. Well, the last collaboration I wanted to talk about is work on the great rivers of Siberia. Here we are on the Ob River. Our Russian colleagues, Max Holmes in the back, I'm over here. Here's Sasha Shiklemanov, the son of Igor Shiklemanov, the very world-famous head of the Hydrologic Institute in St. Petersburg, recently deceased, unfortunately. Sasha Zulidov, our chief logistic person for sampling the great rivers of the Arctic. Eurasian rivers are by far the largest source of fresh water to the Arctic Ocean. On the North American side, we have both the Mackenzie and the Yukon, which are both much smaller than the Ob, Yenisei, and Lena, which are the equivalent in both discharge and area to 3 Mississippi rivers flowing into the Russian shelf. Well, we were interested in the biogeochemical flux and the tracers delivered by rivers to the Arctic Ocean. First, we had to find out, however, if the discharge of these rivers was at steady state, or was it so variable that we had to learn a lot about the relationship between discharge and biogeochemical flux in these various systems. The Russians started in 1936 and measured the flow of all these rivers on a daily basis and had these log books full of river discharge data. We were fortunate to be able to get a hold of it. So what I've plotted here is the river. These are anomalies, that is, deviations from the mean, the long-term mean, for river discharge. You can see the decadal oscillations. This is the black line. And then in 1965 to 1970, this whole system started to turn around. See, the blue line, the temperature had been declining. The North Atlantic Oscillation Index had been declining. Here we had an event in the North Atlantic, or the Great Salinity Anomaly, which was triggered by increasing freshwater fluxes from the Arctic Ocean into the North Atlantic, and started this long period of increasing discharge. Well, this was the first clear illustration in my mind, or one of the first illustrations, that what was happening in all of Siberia, this is the sum of all the Siberian rivers, what was happening is there was a response to global warming, and that's modulated to a great extent by the North Atlantic Oscillation Index, which determines where the water is distributed. As you make the earth warmer, of course, the air holds more moisture, but then you need a distribution system, and when the NAO goes into a sustained positive phase, you'll get a great increase in the high latitudes. We went on later to publish the 2006 paper with other colleagues on what all the freshwater sources in the Arctic were doing over the last 50 years or so. and to compare that to the freshening that's been observed in the Arctic Ocean. So I wanted to give you a little anecdote. My dad spent his entire career keeping tabs on Russian submarines during the Cold War. Some of you can probably remember having nuclear bomb drills where we went down in the basement, huddled under tables. I can remember even as a grade school kid thinking how stupid that was. Fortunately, things have changed a little since then, and I've spent the last 15 years working with Russian, Canadian, U.S. scientists to understand changes in the Arctic, the biogeochemical cycles, and the hydrologic cycle. And to me, this was a heart-warming experience, remarkable and hopeful change in attitudes. But I'm convinced we're going to need a lot more international collaboration to address the needs, to understand what's happening to our world and what we're doing to it. So thanks.